All right. So for this particular lecture, we're going to be talking about stereoisomers of carbohydrates. And by the end, we're going to be able to designate a particular structure as being either the D or the L stereoisomer. First, we got to introduce this concept of chirality. And before we do that, I just want to talk more broadly about this idea of stereoisomers. We've seen this before with regard to our alkenes and how they can either be trans or cis. So the difference between these two structures are the stereochemistry, or their stereoisomers of one another. What designates a stereoisomer is that the connectivity of all the atoms is the same. So the same carbons are bound to the same carbons, bound to the same hydrogens, all that good stuff. It's not like there's bonds that are changed. Different from something like this, which would not be a stereoisomer, but a structural isomer. It has the same formula, but the connectivity of atoms is different. Now we're going to talk about a new type of stereoisomer called a chiral molecule. Okay, so these were cis and trans. That really applies to alkenes. Now we're talking about chirality, which applies both to organic chemistry molecules. We didn't really encounter this concept in organic chemistry, but we could. You would if you were in the 2-semester-long course. But we are going to talk about it because it's really important for sugars. Okay, so a chiral molecule, or a chiral object in general, is an object where its mirror image cannot be superimposed. Okay, so for example, if I took a pair of gloves, they are mirror images of one another, but if you picked up those two gloves, you can't superimpose it, meaning match everything perfectly, right? Versus two glasses, they are mirror images of one another, but they're also superimposable. If you picked up one and put it on top of the other, it would look the exact same, right? So that would be achiral. So this means achiral means mirror images are superimposable. Being chiral means that your mirror image is not superimposable. Okay. So again, we talked about structural isomers. These are molecules that have the same molecular formula, but the connectivity of the atoms is different. So for example, ethanol and dimethyl ether are stereoisomers, but ethanol has carbon, carbon, oxygen, whereas dimethyl ether has carbon, oxygen, carbon. So those two carbons and that oxygen are arranged differently. Same thing with propanol and propanone, right? The connectivity of those atoms are differently. One, the carbonyl carbon's on the end, the other one, it's in the middle. So that's a structural isomer. Stereoisomers are, again, those are when the atoms are all bonded together, but the way that they're arranged in space is different. So you can't superimpose them. Okay, the classic example of a chiral object is your hands. Okay, your hands are perfect mirror, not perfect, but all intents and purposes, perfect mirror images of one another, right? Your left hand is a mirror image of your right hand. But if you take these two objects and you try to put them on top of each other, line them up at your middle finger, they don't superimpose onto one another. They are not sort of the same object in terms of being able to match them up exactly. Okay, molecules have this same sort of feature, the same sort of concept of chirality. So let's take a look at these two molecules here. Okay, they both have the same molecular formula. So ClBrIH. It's a pretty ugly one, but that would be it. Same thing over here, ClBrIH. And they are mirror images of one another. So notice they put this mirror here, and you can see that the left-hand side is the mirror image of the right-hand side. Okay, but if you picked up these two molecules and tried to set them on top of one another, they would not superimpose, right? So if you lined up the hydrogens and you lined up the iodines, the chlorine and the bromines would not match. right? So these, this is an example of a chiral molecule where, or rather this would be a chiral molecule where its mirror image, which is this guy here, is non-superimposable, all right? The pair of them together would be called, so this is a new fancy vocabulary word we're learning, enantiomers, right? So these two together, this is a pair of it's actually like, I always misspell this, enantiomers. Enantiomers. Okay, so an enantiomer is a pair of molecules that are mirror images of one another, but they cannot be superimposed on one another. And it's pretty easy to tell whether a molecule is going to be chiral or not. A chiral molecule will have 4 different atoms or groups bound to a single carbon, right? So the bromine is different from the chlorine, is different from the iodine is different from the hydrogen. That's how I can spot that this is a chiral molecule. Okay. Likewise, you can have more than one chiral centers within a molecule. So for example, if I had something, this is bromine, this is fluorine, this is iodine, and this is chlorine, right? The one thing that I didn't explicitly draw is the hydrogens. So each one of these carbons is bound to 1, 2, 3, 4 different groups. And this carbon is also bound to 1, 2, 3, 4 different groups. Okay. So you can have more than one chiral center in a molecule, but in order to be a chiral molecule, you need to have at least one carbon that's bound to 4 different things. Okay. What would an achiral molecule would be one where at least 2 of those groups match, right? So again, I said that they need to be 4 different things, 4 different bonds in order for that carbon to be chiral. This would be an example of an achiral molecule because you have 1, 2, but then these guys match, right? They're both bromines. And that means that it's achiral, so its mirror image is superimposable. So let's show that real quick. We're going to take the mirror image of this structure. All right, we get that. But now if we just simply rotate that molecule, I think just like 120 degrees, we now get that same molecule. So this could be superimposed onto this and it would match perfectly, right? The hydrogen's pointed up, the chlorine's pointed to the left, and then you have bromines pointed down and to the right. So this would be an achiral molecule. And again, that was easy to spot because it's not, that central carbon is not bound to 4 different groups. Okay, so if it's not chiral, it's achiral. All right, so now it wants to indicate whether or not the red carbon is chiral or achiral. Okay, so is it bound to 4 different groups? So let's look first. This is glycerol. We're going to talk about this a lot when we talk about fatty acids or lipids in general, I guess. But yeah, so this is this molecule glycerol. So looking at that central carbon, this is a unique group. This is a unique group. But then these 2 right here are the same thing. They are both CH2OH groups, right? So I only have 3 different groups bound to that central carbon. So that would make glycerol achiral, right? Meaning it's not chiral. It's not that carbon that's indicated is not bound to 4 different groups. All right, what about in this molecule here? This is MSG. We have that carbon in pink that we're talking about. All right, so this is different. This is different. This is a carboxylic acid group. And then this back here is also its own big ugly thing. Okay, so notice how I'm not just looking at exactly what the next door neighbor is. I'm not looking and saying, oh, this is a carbon and this is a carbon, so those are the same and it's achiral. You look at the whole group, right? So it's not just the atom that's bound, it's the entire group of molecules, and do they match exactly? So this carboxylic acid group is different from this chunk over here, and so that would make this carbon chiral. All right. Chirality is a property both in organic chemistry, but of course we're talking about carbohydrates here. So it's something that's going to be particularly important for sugars. Okay. Before we're going to talk about chirality and sugars, we're going to first talk about Fischer projections. These are a way of representing sugar molecules. You can actually do this for any organic molecule, but it's something that's fairly specific to the study of sugars and carbohydrates. Okay, so for a Fischer projection, you line it up much like we saw before. The difference is that now you have these intersections which represent implied carbons. So much like in our bond line structure, which I've made you guys train over and over and over again this semester, you know that these endpoints and intersections represent carbons. We have the same sort of idea in our Fischer projection where the carbons on these intersections are implied. There's a carbon there. Okay, there's another sort of convention with Fischer projections, which is to keep that carbonyl carbon at the top, right? So if it's an aldehyde, that should be the very first carbon. If it's a ketone, it should be as close to the top as possible. For sugars, we have this special name for the carbonyl carbon. We call it the anomeric carbon. So this is a term, another sort of fancy vocab word that you want to get used to, the anomeric carbon. So this guy right here, and it's just the carbonyl carbon, but again, using this fancy term, is the, anomeric carbon. Okay. The other important thing about sugars is they have a set numbering scheme, right? We always talked about how we're numbering the parent chain in organic chemistry. We have a similar sort of logic in our study of carbohydrates. Sugars are numbered in a very particular way, and that is to give that anomeric carbon the lowest number. So in this case, that would be carbon 1, and then going down the line all the way to carbon 6 in this case, because we're talking about glucose. Okay, the last feature of this Fischer projection is each one of these carbons for our sugar are chiral. Right? You can convince yourself that they are all bound to different groups. It's always a hydroxyl group on one side and a hydrogen on the other side, but then the carbons that they're attached to are also different, right? So these are all chiral carbons. The last one doesn't get that same sort of fancy skeleton-like structure because it's an achiral carbon. So it's just left as CH2OH. So the last carbon is always achiral. And so it's usually not included in the structure. I will say I've seen on an ACS exam them try to be tricky and show you something like this. H, H, OH. Typically though, that's a kind of a weird format. You almost never see that. You will always see it as CH2OH. And that's again, that last achiral carbon. Some features of Fischer projections. Okay, so just this isn't different from what we talked about before, but this idea that if this central carbon has two groups that are the same, so in this case, these two methyl groups are the same, that's going to be achiral. And that's what we talked about before, right? In order to be chiral, you have to be bound to 4 different groups. Now, this is a little bit different. And I'm not going to teach you guys how to go from like the bond line structures to the Fischer projections, because frankly, it's kind of a pain in the butt and it's not going to be something you're going to need to do. Trust me that there is a way to do it. Instead, we're going to just be talking about features of Fischer projections so we know how to use them. You're not going to be expected to jump back and forth between different representations, okay? Okay, so if two groups on a Fischer projection are swapped, and they're almost always going to be in this horizontal bar here where they're swapped, although that it doesn't really matter which two positions. Nonetheless, if those two positions are swapped, then that produces the mirror image, right? So these two molecules here are mirror images of one another. Does anybody remember the fancy name that we put on that? For that pair of molecules? This would be a pair of enantiomers. Right? Remember, enantiomers were that special vocab word we had for a pair of mirror images. So we can spot a pair of enantiomers in our Fischer projection if any two groups are swapped. And again, it's almost always going to be these middle two here, but nonetheless, it could be any of them. Okay, the last thing that's important for Fischer projections is if you rotate any three groups, so hold one group still and then rotate the position of the three other groups, that's actually all the same molecule. You've just given a different representation of an identical molecule. This isn't the enantiomer or mirror image or any of that sort of stuff. All you're doing is sort of changing the representation. So this is also going to be something that's important later. So rotating three groups means that you have the same molecule. And so as I sort of said, we're going to use these Fischer projections, and it's going to make it really easy to spot those chiral carbons because they're going to be sort of those implied carbons. So here we have erythrose is one particular sugar. This would be a, let's just make sure we can do this, right? So it's got 1, 2, 3, 4 carbons and an aldehyde group. So this is now a new aldotetrose. They all have their own names. You're not expected to know them, but you will be given problems like here's this sugar you've never seen before, right? So figure out, like, classify it. That's really the only big thing that we've done up until this point, but we're going to learn some other fancy stuff that we can do given these structures. So here we have this structure. This is how we classify enantiomers of a particular sugar, is as either D or L stereoisomers. Okay, so this is L erythrose. How do we determine whether it's D or L? We look at the last chiral carbon and we look at where the OH group is in our Fischer projection. If it lies on the left, as it does here, that's the L isomer. If it lies on the right, that would be the D isomer. Okay, so this image here is the L isomer of erythrose. Now, what it wants us to do is to give the enantiomer, or the mirror image, which would be D erythrose. Remember that in order to produce the mirror image, we're going to have to swap the positions of all those horizontal images. So in this case, basically, these two groups are going to be swapped to produce the mirror image, and these two groups are going to be swapped to produce our mirror image. So D erythrose would look like this. And again, we can spot it as the D isomer because now that OH group on that last chiral carbon is on the right. Okay, how are you going to remember the DL isomers? Well, L is on the left. All right, again, only talking about that last chiral carbon. So that's why it's so important that you stack up it up in the correct way where the aldehyde or ketone is closest to the top. Okay, enantiomers in our body. So for the most part, an enantiomer will have the same physical properties of, two enantiomers will have the same physical properties. So the same boiling point, the same melting point, viscosity, all those other sort of physical properties, molecular weight, all that sort of stuff will all be the same. However, your body, just by the way that our body works, and we'll talk about this a little bit more when we talk about enzymes, but your body works by what's called like this lock and key sort of, or the enzymes in your body work by this lock and key kind of signal, where basically your enzymes in your body have this perfectly fit little pocket and a molecule plugs right in. So if it's the mirror image, it's not necessarily going to fit the same way. And so we can interpret enantiomers of the same compound differently. So a perfect example would be the difference between the enantiomers of carvone, which is made by the spearmint plant. One of them tastes and smells like spearmint. We all know spearmint. And the other one, the other mirror image of that molecule, has the taste of caraway, which is like the rye and rye bread that you taste. So very different. And these are just mirror images of one another, right? Same sort of molecule, but they're mirror images, and those mirror images are not superimposed. They're enantiomers rather, and your body interprets them in terms of taste and smell differently. All right, so classify each of these as either chiral or achiral, okay? So whatever I'm looking for a chiral molecule, what am I looking for? For that central carbon to be bound to 4 different groups. And so this is 1, this is 2, this is different, and lastly, this is different as well. So this is indeed a chiral molecule. Likewise, this is different, this is different, this one's different, and then this one's different. So this is also a chiral molecule. Let's do another problem. How would you draw the enantiomer of this molecule here? Remember, you're going to swap those 2 groups. So Cl, C, Br, we're going to keep these the same. We're going to swap the groups on that horizontal axis. So this would be a pair of enantiomers. All right, and again, I swapped which side these 2 groups were on. So determine whether the following pairs of Fischer projections can or cannot be superimposed. Remember, if they cannot be superimposed, that's another word for an enantiomer, right? So here, let's see what's the same. This group is at the top here, it's at the top here. This group is at the bottom, at the bottom. This methyl group is on the right over here and on the left over here. And this hydrogen's on the left structure and the right on the right structure. So these would be a pair of enantiomers which cannot be superimposed. Okay. What about the next one? Well, so I have my bromine here, my bromine here. My methyl group has seemingly swapped sides on one side compared to the other. Same with this hydrogen has swapped sides on one side compared to the other. However, there's 2 hydrogens. So this is not a chiral center, which means that this molecule is achiral and indeed can be superimposed. They're actually just different representations of the same molecule, right? And we can tell that right off the bat that they're not going to have any weird business about being superimposed because it was achiral. Because right off the bat, we could tell that these carbons were not bound to 4 different groups. All right, so now we kind of like mixed in some organic chemistry where we're just talking about this concept of chirality, but remember the focus of this chapter is sugars. So one of the big skills that we need from this lecture is to be able to classify a given sugar as being either the D or the L stereoisomer. And again, how are we going to do that? We're going to look at the last chiral carbon, so that would be this one here. And we're going to see whether the OH group is on the left or the right. If it's on the left, it's the L isomer. Since it's on the right, this would be the D isomer. Okay. One other thing that we can do just to make sure that we understand is, what if I said, okay, great, that's the D isomer, draw me the L isomer. Remember, you're going to need to flip all of those chiral centers, going to need to rotate, swap those groups. So the top stays the same. The bottom, our last achiral carbon, we're not going to draw, or we're not going to represent in this skeletal structure. But all the other chiral carbons, we need to swap the hydroxyl group and the hydrogen. So now the hydrogen's on this side and the OH is on this side. OH, H, OH, H, H, OH. Okay, so that's how I would get the enantiomer or the L isomer of whatever sugar this is. It didn't even give us the name of it, but nonetheless, we could find out that information. Again, we can convince ourselves that this is indeed the L isomer because that hydroxyl group on the last chiral carbon is on the left. Okay, and notice how, we talked about for Fischer projections, you swap these two groups. Notice how these two structures are indeed the mirror images of one another, right? If I had a mirror and I held it up to this page, I would indeed get a drawing that looked just like this. Okay. Yeah, this is all pretty straightforward. Again, we just need to be able to tell whether or not we have the D or the L isomer of this particular sugar. So actually take a second and classify which one's the D isomer and which one's the L isomer. Remember, all we're going to do, this is a really easy example because there's only one chiral carbon, but you would look at that last chiral carbon. Since this one has its OH group on the left, this is the L isomer. And since this OH group is on the right, that would be the D isomer. Again, we sort of stressed this a bunch before, but nonetheless, you're only looking at the chiral carbon that's the furthest away from the carbonyl group, right? So the furthest to the bottom. Again, you ignore that last one, right? This is actually the last carbon, but it's not a chiral carbon. So we're not going to pay attention to that one. We pay attention to the last chiral carbon. That's how we get our D and L designation. Again, L means it's on the left. D means it's on the right. Okay, so here's a molecule of glucose. Is it the D or the L isomer? Well, I'm going to look at that last chiral carbon. Since the OH group is on the left on that last chiral carbon, this would be the L isomer. Remember, we have that numbering scheme where we start at the top, start at that aldehyde carbon, and then go down. Now, how would I get the mirror image of this? Right, so I would start by putting my carbons that don't have any sort of chirality, the ones that I'm not worried about, would be put in there. Make sure I have the correct number of lines. So 1, 2, 3, 4. And then I need to swap each one of the groups on these chiral carbons. So for L-glucose, it's the first, or the OH group on carbon 2 is on the left. That means the OH group on carbon 2 needs to be on the right. All right, so you're just swapping for each one of these carbons going down. And so this would be D-glucose. Okay. Again, just to kind of drive home this point, circle all my OH groups so you can see that they indeed have switched sides on the chiral carbons. Okay, so these are the two isomers of glucose. Again, we designate them as the L or the D isomer based on the position of the hydroxyl group on that last chiral carbon. Would you have known that this was glucose without being told it? Well, I mean, it is on the constants page, so you could have seen it that way. But in general, again, your goal is not to memorize glucose looks like this, galactose looks like this. It's to be able to analyze these structures when you're given one. Okay, these are the three most important ones. Okay, they're all hexoses. Glucose and galactose are aldohexoses, whereas fructose is a ketohexose. So let's look and see if we can spot the difference between glucose and galactose. Notice that it is just the position, or rather the stereochemistry of your fourth carbon. Right, so in D-glucose, the hydroxyl group on the fourth carbon is on the left or on the right, whereas in D-galactose, the hydroxyl group is on the left on the fourth carbon. So that's the only difference. In general, that's sort of how, that's the difference between all sugars. They can differ by the number of carbons. They can differ by being either aldoses or ketoses. But then if you're talking about all the, for example, aldohexoses that exist, the difference is going to be where the hydroxyl group is, right? So if I took this third carbon and I swapped the hydroxyl group, this would now be a new sugar. I honestly don't even know what sugar that would be, but it would be another sugar. It would have another name, maldose, mannose, something like that, okay? So that's the big difference between all the different sugars, is just the position of the hydroxyl group on those various carbons. Fructose again is a little bit different because it's a ketose, but these are the three most important monosaccharides and the one we're going to see over and over again. You don't have to have their structure memorized. You have to have memorized that they're monosaccharides. And when we start talking about disaccharides, you're going to want to know, like, for example, sucrose is fructose and glucose. So that's another thing, that's another component of stuff we just got to know. So talking about them a little bit individually. So D-glucose, this is by far the most common one. It corresponds to the sugar that is found just about everywhere. So in photosynthesis, plants generate glucose. Your body, a lot of the times when your body's metabolizing other sugars, what it does is first turn them into glucose and then go through a normal metabolism cycle. So glucose is the most common one. D-glucose is also called dextrose. So if you look on the back of like an ingredients list for a food item and it says dextrose, that's really D-glucose, right? There's also this L-glucose isomer. We don't really have that in our bodies, actually. So dextrose and D-glucose are the same thing. And that's what our blood sugar is as well. So we're going to talk about maintaining certain blood sugar levels. What we're talking about is the concentration of glucose that's in our blood. And we're going to see it's the building block for a bunch of different disaccharides, sucrose, as well as lactose, as well as maltose, as well as a bunch of polysaccharides. So cellulose, glycogen, amylose, amylopectin, all of those contain glucose as their building block. D-galactose. D-galactose is another important one, mainly because it's the other component of the disaccharide lactose. So it's an important sugar for that reason. It's also important in sort of cellular membranes in the brain and the nervous system. It plays an outsized role as kind of the sugar of the brain. And then fructose is another very important one. It gets its name, it's a big component of fruit. Sucrose, which is table sugar, is a combination of glucose and fructose. Fructose tastes sweeter. Your tongue detects it as sweeter than the other sugars. And so that's why you have high fructose corn syrup, which is syrup derived from corn that contains a large amount of fructose. And that's additive to candies and all that sorts of stuff to give it that really, really sweet taste. All right. Again, one of the biggest skills we got from this lecture, first of all, being able to identify those chiral centers. We had those vocabulary words, stereoisomer, enantiomer, chiral, achiral. But then with regard to carbohydrates, the big thing was to be able to identify whether it's the D or the L isomer and then draw the enantiomer of it, right? So for this is ribulose. Do we have D ribulose or L ribulose here, right? So what are we going to do? We're going to look at that last chiral carbon. In this case, it is on the right. The hydroxyl group is on the right. So this would be D ribulose. If I wanted to draw L ribulose, this would be a good idea to pause and try to do this on your own and then play it back. Again, first thing you're going to do is lay down all the achiral carbons. Those ones aren't the hard ones. So in this case, this carbon at the top is achiral, this ketone carbon's achiral, and then down at the bottom, that carbon's achiral. For our other two carbons, now we got to swap the positions of the hydroxyl groups and the hydrogens. So this would be the structure for L-ribulose. All right, and there are two, your body goes, the human body goes way far out of its way to try to maintain a constant blood sugar level at all times, right? So right after you eat, it might spike a tiny bit, but then it will sort of plateau out, and your body wants to keep it within this very narrow range. There are two different diseases that lead to either having elevated levels of blood sugar or low levels of blood sugar. The first one is called hyperglycemia, hyper meaning more. This is diabetes. And the other one is hypoglycemia. It's just called hypoglycemia, and that's lower than normal blood sugar levels. So diabetes occurs because your body doesn't regulate its blood sugar concentration. A key player in regulating that concentration is insulin. We're not going to talk about how it does it, but bottom line is insulin is used to regulate your blood sugar levels. Diabetes is slowly very bad for you in that it slowly causes damage to your cells over time, having that increased level of blood sugar. Excessive thirst and irritation messes with your appetite a whole bunch. In general, it's just very bad for your cells, and it's something that's not detected, always detected very quickly, because it's like slowly your body just feels more and more sluggish. The other extreme would be hypoglycemia, and this is something that's a little bit easier to detect when your blood sugar levels are low, because you basically just don't have any energy at all, right? So this is actually now the opposite effect, where there's an overproduction of insulin. And again, we're not going to go into details about how insulin works, but it's important in regulating blood sugar levels. But so you don't have any sugar floating around in your blood, any energy for your cells. So this leads to symptoms such as weakness and muscle tremors, as well as just dizziness. And yeah,